Today the SRC committee is going to present our new rules book, the Guidelines for Science and Engineering Fair 2020 to 2021. I'm Susan Appel, a member of the SRC from New York, former teacher, school administrator, with much local science fair experience. And I'd like to introduce my colleague, Henry Diston. Tell us something about yourself, Henry.
Can you hear me?
Yes.
Yeah, I'm Henry Diston. I run the Delaware Valley Science Fairs out of Drexel University in Philadelphia.
And Joe Scott? Joe?
Hi, this is Joe Scott. I'm a teacher at Satellite High School in Satellite Beach, Florida. And I have about 60 or 70 students every year that are involved in the regional science fairs.
And Michelle Glidden, who is working for SST and supposed to be hosting this, but we can. Michelle, can you hear us?
Yes, thank you, Susan, and I apologize
okay. So you're in
for my technological problems here in the beginning. But welcome to everyone, and thank you for joining us today. We're here to talk about the international rules for pre-collegiate research, the guidelines for science and engineering fairs that have been written for the 2020-2021 school year. And so we've just published them today, as is our tradition at the Regeneron International Science and Engineering Fair. And we're here today to talk a little bit about the work of this committee and primarily about the rules and their purposes and how you can be sure that you're using them in your process as we move forward. So as they've each introduced themselves, they are volunteers, Henry, Joe, and Susan, are volunteers for the society as members of the Scientific Review Committee, and they have several other colleagues who work with them throughout the year. And their two core purposes are to, first and foremost, to review the international rules annually and to make revisions as we move forward and we reflect on the questions and the projects that we see in any given year. They also answer an email account, src@societyforscience.org, which is a helpline, honestly, throughout the year for students looking to do research, to their educators, to their parents, to their fair directors, to aid in the process. And then especially they spend the spring reviewing the projects of the Regeneron ISEF to ensure that they are in compliance with the rules in order for competition. For this year, in our virtual experience and without judging, their role has been to review and approve those projects for publication. And so if you see the project in the finalist exhibit hall, it means that it did go through SRC review. But to go ahead and move us forward, we'll take a look at the rules and their purposes. And so Henry, if you don't mind starting us off.
Well, as you can see on the screen, the purpose of the rules, I won't read all of them, but basically these rules are all about risk, and we want to ensure that the presenters and the researchers are complying with these regulations for the sole purpose to assess the risks and to be able to safely do their research. It's also, and I say this a lot to the kids, this is exactly what working scientists have to go through also. There is always oversight. And so it's the same thing that you have to go through. We want to make sure if you're dealing with certain projects, research, particularly with human participants, there's oversight, and the same with vertebrate animals and also with potentially hazardous biological agents. Anything else, Michelle? Anybody else want to chime in?
Yeah, I have a few things I want to add. One of the things that as a teacher, and I'm sure all the teachers listening there know exactly what I'm talking about, is we get to work with these really bright kids. They're verbal, they're curious, they are just a joy to be around, but they are still kids. And we need to remember that this is pre-collegiate research. Our goal is to make sure they don't hurt themselves or hurt anybody else, which is why adhering to the rules is so important.
Thank you, Joe. And then to go to the next slide, Susan, will you walk us through a bit of this?
Gone through the rules with us may remember our former ethics statement. And until 2 years ago, it started with the sentence, scientific fraud and misconduct are not condoned at any level of research competition. And then we it's the negative. And it's sort of saying what a teacher says, bad behavior will not be tolerated. So we decided to do what good teachers do and start with something positive, provide a roadmap for the type of behavior that we expect, and which led to our current statement, which includes a description of the highest ethical standards in the 8 categories on this page. However, we also have a discussion of consequences for scientific fraud and misconduct, and that you will find in the last paragraph of the ethics statement. Thank you.
And then we come into a few things that are changes for next year and clarifications, particularly in this past spring, where we know 1/3 of our fairs were not able to hold competition. And so obviously in preparation for those fairs, many students had begun research. And so the question has come to us already from several factors. And so we just wanted to clarify in this year's rule book and then in this session today that the eligibility requirements remain the same as has been typical. And that is that a project may extend for 12 months of continuous research. And the earliest start date for that research is January 2020 for the May 2021 competition. And so that means that if you began your science fair project for the spring in 2019, that is this year's project and cannot be considered a project for next year's round of affiliated fair competition and potential advancement to the Regeneron International Science and Engineering Fair. And we just want to provide that clarity. Obviously, students are encouraged to take the research that they began and to do a valid continuation. And we're happy to provide advice about that during this session or obviously in the SRC email account post this session. But we did just want to clarify that for the confusion there. The competing in the science fair is not the only condition upon which a project can't advance. Now, if a student happened to not have started their project until January of 2020, and they only work on it for 12 months, then it could be considered eligible for competition next spring. So I just, we raise that awareness because we want to be sure that students recognize the rules of eligibility. And so I'll move to the next slide. Oh, Henry, you have a comment to add, please.
A continuation for those of you that have done work for this year, that we're hoping to enter for this year, I strongly encourage to continue your work as a continuation project. There are some students that have some idea that continuations are bad, but actually that's what science and research is all about. You learn from what you've done, and from what you've learned, continue to do your research and answer some of those questions. So I strongly encourage those that weren't able to participate in a fair this year with their research to continue that research and do some extra ones and extra work on that. And that would certainly be fine. And like I said, continuations are actually good things. So don't look at it as in a negative light. That's all I have to say.
Thank you, Henry. And Joe, now if you'll start us off on this next slide for us.
Okay. Every year, the single biggest problem area tends to be research involving human participants. And that's no exception for this year. This year we had quite a few problem projects involving humans. The rules are relatively straightforward, and it basically requires that first off you obtain an institutional review board review and approval. Now we know that that can be a problem in the smaller areas or no, smaller fares in the rural areas, but that seems to be an area that we continue to have problems with. So when you're constituting your IRB, please make sure that you try and get the members as close to the project as concerned. If you're having a student that's doing a psychological project and your medical expert is a school nurse, that's probably inappropriate. You would be much more likely to have a psychological person on your IRB that would understand the risks that are involved and inherent throughout there. And the other thing, well, two other areas that we run into problems with. One of them is if your student is working at an RRI, your local IRB will not be able to approve it. You're going to need the institution's IRB to approve that project. And the last one, and this is one that we really want to stress, is we need parental permission when your students are working with minors. And that all too often is not happening. People are assuming that it's okay because it's not a particularly dangerous project or not particularly concerning as far as psychological risks are concerned. But we believe that parents have the right to know what their students are getting involved with. So parental permission is something we want to make sure that we really focus on in this next year. Thank you.
Henry, go ahead and cover the next two lines, if you will.
We talked about research done within the timeline. The size of team projects. Now, team projects have a maximum of 3, and those 3 have to be involved in the whole process. There have been quite a few times that a team of maybe more than 3, say 5 or 7, would do a project, and then they would designate 3 people from that team to present at ISEF. That is not allowed. The team can only be 3 people, and those 3 people have to be involved with that project all the way through. One of the areas that we see this is in the robotics competition first, which is a team of much more than 3, and sometimes they would designate 1 or 2 people to represent that team. Again, that is not allowed. It has to be those 3, and those 3 have to go through the whole thing, and that's it. So hopefully that clarifies that a bit.
Thank you, Henry. And Susan, will you cover the potentially hazardous biological agents for us, please?
Sure. We're one of our biggest problems with the PHBAs is that some students try to culture bacteria at home, and this is prohibited at all levels, even if it's even if it's something that is not hazardous, a BSL 1 project. Also, another big issue has been students doing BSL 2 projects in a BSL 1 lab. So you have to make sure that the students have the proper protection and the proper supervision on any of these projects. Thank you.
Thank you, Susan. And now I want to cover a little bit about some of the changes to the rules book this year, and these will really only be significant to those who perhaps who know the rules book particularly well. We did not make significant changes, so that's kind of the good news for everyone. But there are 3 minor things that I just wanted to point out. One is that we did continue to provide minor clarifications of language. That's something that we look at very closely every year. There was also an exempt tissue that was included, which is human capillary and blood collection. So essentially a simple finger stick, which in the language of our rules previously would have required a lot of permissions and supervision. And so we've added that to tissue exempt from that level of review. The other thing that we added is really a lot of work that the Society has been doing with the Lemelson Foundation around invention, and recognizing that so many of our students working and doing science fair projects are also doing engineering and invention projects. And our rules have been very biologically focused just because of so many of the protections around animals and humans that are natural. But there are often invention projects that engage and involve with humans, potentially animals, and that do require their own considerations of safety and proper review and approval. And so we have added a reference in the book to begin to address some of the questions that a young inventor should look at to help improve and to be sure that they're in compliance with the rules. So we encourage folks to look at that new resource and provide us feedback as we move forward. And then more in terms of the housekeeping of the ISEF SRC and the Society, we are forever looking to improve. And one of the things that we have been exploring for the last couple of years and would love feedback from the audience here and throughout the school year as you explore the rules and you use them as a resource, and that is the organization of the book. It's a long book. There's a lot of detail in there, and it comes down to that there are about 4 elements of material in the book, and it's a question of whether a reorganization of those 4 elements would be helpful for the end user. Right now, the book is really in discipline, right? So we have a human participant section, we have a vertebrate animal section, we have a potentially hazardous and biological section. We have a hazardous devices section, but each section has rules for competition and for the way you conduct research. There are rules specific to competition at Regeneron ISEF. There is then the required documentation and the forms at the back of the book, and then there is a lot of guidance for sound and safe research projects that we continually add to, but they're not technically rules. And so it is just a question as we look forward to the organization of this book, how can it be the best resource? So with that, what we really wanted to spend today's session doing is answering your questions. I will remind everyone, and I think I said it at the top of the session, we are available to you through the SRC at societyforscience.org email account. We encourage questions at the beginning of your research journey so that we can help guide you on the right safe pathway. Many projects require pre-approval. They require risk assessment. There are some things that you can do to ensure that you set up the project. Often the quote-unquote pre-doing is a longer process and often more involved than the actual experimentation phase. And it's important to consider that as you begin your next project. Also, here is a link to our international rules and guidelines for 2020-2021, and so we encourage you there. But without further ado, let me take a look at a few of the questions and pose some to the panelists. So here's a question, and just raise your hand if you want to be the one who answers it. When students do research with their advisor, often advisors give very big hints or even solutions of the research until what level of advice would not affect the results of students' research. So what is the balance of a mentor's involvement in the project? See, any thoughts around that?
I could give us a stab there, and maybe Joe, being a teacher, could help a little bit. But yeah, quite a few students are lucky enough to get mentors, not necessarily go to a laboratory or whatever, and receive guidance from those mentors. Obviously, you don't want to have the mentor do any of your work or a lot of your research. This is one of the important aspects of the interview. We can tell pretty quick who did the project, and how much the researcher really knows about that, and then how much it was for the mentor. So my advice to students is it's your work. Get guidance from the mentor, but you do the work and make sure you understand what you're doing.
Thank you, Henry.
The only thing I would add to that is that oftentimes mentors, again, they're typically college professors, and they're not used to working with pre-collegiate students. So I think it's always important that the adult sponsor or the student's teacher be involved in that to make sure that the mentor understands the mentor's role and doesn't take on the aspect of just assuming that the student is another grad student.
Thank you, Joe. And while you have the floor, here's a human participants question for you that you can start us off with. When students use family members to survey or use them as test subjects, should they go through an IRB and use permission forms, or is Form 3 sufficient?
No, Form 3 is not sufficient when they're using family members for part of their project. They need to go through an IRB and go ahead and do the regular consent forms. The only time that it is agreeable for one family member is if you're in a situation where you have a student who's doing something that he cannot do or she cannot do. For instance, it requires driving a car and they don't have a license. Then the family member can be involved as the driver, but they're only helping the project. They're not part of the project.
Thank you, Joe. Susan, a question for you. Have we established any rules related to COVID-19? I know we have not in this year's rules book, but I know you had some thoughts around that.
Yeah, so this comes under the category of good news, bad news. And it turns out that the COVID-19 is BSL 3. So any kind of project where the student is working directly with this organism is prohibited and should be. As we know, this is very dangerous. So there are projects that can involve COVID-19. You could use databases. You can do behavioral projects. I think you could probably find projects in almost any category, except maybe microbiology, wet labs. But you can find many projects related to that, and I expect that we will next year. But please do not even consider working with this organism.
Thank you, Susan. A related question that perhaps Susan can start us off, and then maybe Henry and Joe, you might also have some thoughts around. And that is a question around how can you differentiate between essentially a PHBA that needs SRC pre-approval and one that only needs a risk assessment form. They also raised the issue of the fact that given that COVID-19 will restrict and limit students in the laboratories this year, what is allowed to be done in a home laboratory environment with parental supervision?
Okay, so in a home laboratory with parental supervision, there can be no culturing of any bacteria, no matter how harmless it may seem. You can do a fermentation project, and that's okay, but you cannot do any kind of culturing. So you will be limited as to what can be done at home. I suggest, and it's something I'm going to be doing tomorrow, the Regeneron virtual ISEF is having workshops on what can be done at home. And so there are some projects in the life sciences that can be done at home, some projects using big data and so on. Also with how do you know that whether a project is a PHBA and needs a Form 6A or whether it just needs a hazardous chemicals or hazardous devices forms of Form 3. Our rules book has some information on it. Also, when I look at a project, my resource is ATCC. And even though I think I know what some of these, what the biosafety level of some of these organisms it would be, I don't always know, and I always make sure that I check on the ATCC website, the American Type Culture Collection, and that will tell you the biosafety level and give you an idea of what can be done where.
On to that, to the question, there are some studies that can be done at home, PHBAs that are exempt, and that's in the rule book at the end. One of them would be slime molds. We see quite a few students working with it. That is exempt, and we don't consider that a PHBA. Also, E. coli K12 is also exempt. So they can be done at the home. We do require a risk assessment for those, but because they're relatively harmless, we have made them exempt, and that's at the end of the section in the rules book. So if you're not sure, you can go to the rules book, particularly the end, and we mention what's exempt and what's not.
Thank you, Henry. And while you have the floor, here's a question about continuation projects. Can you do a continuation project over multiple years, or can you only continue after 1 year?
You can continue over multiple years. And we see quite a few of that. It would be phase 1, phase 2, phase 3, phase 4. And we kind of encourage that. As I mentioned, that's what science is all about. However, you are only judged on this year's work only, and not what you did before. But what you did before, you learned a lot, and so you're able to apply that to this year. But the judges are just looking for work this year only, and that's it.
Thank you. Joe, here's a question for you. How does a student go about getting their research approved by an IRB if doing their research on their own this summer or fall due to the inability to be in a lab or research institution?
They can do it virtually. I mean, you're still going to have to get an IRB, and I realize that the COVID-19 has made everything like that very difficult, but there's no reason why they can't email their school principal to start the process. Local IRBs are not as difficult as we like to make out putting them together. I do know it is hard to get that medical or psychological professional, but as far as the teacher and the school administrator, those should be relatively easy to get through email. And then get with your regional fair director, and possibly you may have a regional IRB that may be able to work with it, but I know it does present a lot of challenges. I don't know if that's a good answer or not, but it's something we still need to do.
Yeah, and I will say it's not an unusual problem for any student exploring during the summer. Obviously, the limitations this year in not being able to work through the institution, but the school IRB is, to your point, a body of individuals that can review your project even if you're not in person. And I expect that your research teacher can help form that group with you virtually and ensure that you get the proper pre-approvals and review prior to the beginning of experimentation. Here's another question that we have that someone asked for us to clarify again what is the new exempt tissue, and that is the capillary human blood or the finger stick. And so here's a related question. With the finger stick for blood being exempt, if a student does a blood sugar study, i.e. how diet affects my blood sugar, which forms approvals would be required? Susan, would you like to start us off?
Sure. So for sure you don't need a 6A or a 6B. And if you are in a research institution, you will need a 1C. We would suggest that you have either a qualified scientist or a designated supervisor who has some knowledge and can help if there's a problem. So you would want probably a Form 3. And if you are, of course, doing this with other people, then you have to go through an IRB.
You're not using anybody else, then you do not need IRB approval. But as soon as
Right.
you get somebody else involved, then you do.
Thank you, Henry. Right. If you, the researcher, are the subject. This is also just a clarification. There's a follow-up question around culturing at home and whether any of it can be done. And the answer is the rule for all projects involving any microbiology or culturing essentially is that it is not allowed in a home environment. That is different from collecting samples from the environment. And so here's a question, and I'll let Susan perhaps start us off. Am I correct in that students can take swabs or samples at home or in the field and immediately transport them to an appropriate lab for the actual culturing process under the necessary lab conditions at the appropriate biosafety level?
Exactly. The short answer to that is yes. The same rule does apply to collecting samples.
Right now, right, and in today's environment, you might consider whether that's a safe practice with the concerns of unknown materials and the potential of COVID-19 being on surfaces, whether it's smart for your students to be going around swabbing for unknowns. But in terms of our rules, those rules have not changed. You cannot keep the plates at home, but if you collect and immediately move them to a proper laboratory environment, that can be done if the plates are not opened. Go ahead, Henry.
Right, collect and seal.
Well, yeah, you said it, because those plates, once you swab it on those plates and it's unknown, those plates have to remain sealed the entire time. And then when you're done, I would take pictures or whatever, but then has to be disposed of, bucket of chlorine or whatever, but you cannot open those plates.
Here's a question for you, Joe, and that is, where can we find guidelines on the types of materials or documents to put into an engineering project?
Well, I think the first place to look is, of course, is in the rules. Also, the new guidelines that you put together. Michelle did most of the work on the new guidelines that you put together. That helps a lot. So it all comes down ultimately to risk. And you need to be, again, taking a look at the pre-collegiate nature and making sure your student is not putting themselves in a situation where they can harm themselves or harm somebody else. So as long as you're taking those things into effect, probably going to need a Form 3, a risk assessment, and that should probably take care of it.
I think that's right. The generally engineering projects, math projects, sometimes physics projects, the projects for the rules for all projects where there are not specific areas of concern. Many engineering projects fall under those lines. I think where we're looking to provide clarity to students and teachers is that if they build a prototype and they want to then test it with humans, or they want to survey and create the user case. And so then they've asked for opinions and collected personal information from end users. Then they've, again, kind of dipped into the human participant rules and surveying of individuals. And so often an engineering project where they think they're kind of clear of some of the trickier rules, they need to just be aware and ask some of those key questions early about how they intend to proceed and whether they will end up working with anything hazardous that requires designated supervision or humans or PHBAs or vertebrates. Absolutely. Let's see if there's another question for everybody. Right. So here's a question along those same lines. Our local fair is recommending that all students provide a risk assessment form for every project as best practice. Has the SRC considered requiring this form for everyone? Henry, I'll let you start this off because I know this has been part of our internal conversation every time we meet.
Yes, I mean, obviously the rules, as we mentioned right in the beginning, is all about risk. However, to require all projects to have a risk assessment form might seem kind of silly to some, particularly if it's a mathematics project and you're trying to solve a theorem, or if it's a physics project and you're looking at different objects in the sky, different galaxies and stuff like that. There's obviously no risk. One of the things that we're very cognizant of is we don't want to overwhelm teachers and students with a lot of forms, and requiring a risk assessment form for projects that really don't need one kind of dilutes that a little bit. We want to kind of cut down on the form. So that's the answer. The other question that comes in, a lot of people saying, should risk assessment forms require prior approval? You certainly can do that. The local fairs and the affiliated fairs can have stricter rules if they want. And I suggest if you're very concerned about that, then you can maybe put that through a prior approval process. But again, there are some projects that really don't need to have a risk assessment form because it clearly is no risk.
Yes, please, Susan.
Yeah, also, I'm looking at the Form 1, the checklist for adult sponsor. And one of the things, the adult sponsor is supposed to go over this with the student at the beginning and determine what forms are necessary. And one of the things is they are looking to see if there is any risk. And under other, right at the bottom of the page, it says Risk Assessment Form 3. So, and that's one of the things, one of the reasons we have not required it for everybody is because the adult sponsor is already supposed to look at what kind of risk there is.
Joe, any thoughts about this topic before we move on?
The risk assessment can oftentimes be burdensome if we are doing it for every little thing. You know, in our fairs, we work with middle schoolers and with high schoolers. And a middle school kid working with a power drill is certainly something I'd want to see a risk assessment on. But a high school kid with that same power drill probably should not be a concern. So you need to take a look at the kids involved, the individuals, and what that risk is. And that needs to start with the adult sponsor and then the student's teacher.
Great, thank you. There's a question here that speaks back to the eligibility and the situation that we have this year with so many students who were not able to compete in their fair. So can you clarify again, students that were not able to compete this year and will be continuing their data collection must indicate they are collecting new data. So just to again clarify, if a student began their project before January of 2020, so this past January, then that project is considered last year's project for next year's school year, even if they were not able to compete with that project. If they began the project January 1, 2020 or after that, they can work on that project for the next 12 months, and whatever pre-approvals and processes that they went through to begin that research would be valid for competition in the spring of 2021, presuming that's when their first science fair opportunity is. And we did this because there are lots of students who obviously did compete at some level. We've always had kind of an 18-month rule, 12 months within 18 months. And to try to extend that and change that time period just did not seem to make sense. So hopefully that's clear to folks. And a valid continuation, as Henry expressed earlier in this session, is not a bad thing. Often judges and experts in the field appreciate someone's persistence in working on a problem realizing the next step in that journey and pursuing it. So it's not that if you're within the allowable 12 months that you need to have a new process. It's that really you should be thinking of it as next year's project, and then it needs to have the elements of a valid continuation. Now, there is also a question here about the difference between a valid continuation and a longitudinal study. Would somebody like to address that issue?
Okay, Joe, go ahead.
The main difference is what's the variable. In a longitudinal study, the variable is time. So you're looking at the same situation over a period of years and seeing if there's any change in that situation over time. For instance, here in my county, the Indian River Lagoon is something a lot of our students look at. And they check to see if, say, over time or with some kind of a new project, has the water quality changed. That would be a longitudinal study. While a normal continuation study is looking at changing an element of their project from the previous year. For instance, if they were studying a CRISPR technique one year and the next year we're looking at a different genetic engineering technique, that would probably be a continuation project.
Thank you, Joe. And we're coming up on time, so perhaps one of the ending questions for each of you to reflect on, and that is, obviously we're at a virtual ISEF this year, and there have been a couple of questions around digital and paperless processes. And so talk to us a little bit about the digital signature and how fairs should follow through on that. And talk about what we've seen over the last couple of years from fairs.
Well, I could. We do allow digital signatures on a lot of the forms, but to proceed, we just got done a virtual fair, and who knows what's going to be happening next year. But I would suggest that even logbooks and things could be digitized. And students should consider that when they're working with their materials or whatever, to make sure you can have everything electronic. Therefore, if you are going to have to do a virtual fair, at least you have that and you can just upload those into a folder or however those fairs are going to deal with that so the judges can look at it.
And a final question is from a middle school student. What is the acceptable level of middle school projects documentation? Do you expect middle school projects to fill out the same forms? I also know that we have this debate annually. And so, Susan, will you start us off, and maybe we'll have last thoughts around that topic and then conclude for the session.
Sure. I know in the Broadcom competition this is an issue, and also it seems to be there seems to be no definite decision thus far on what forms should be used for the middle school, and I know this has been also under ongoing discussion, and I think I have no information other than that.
Michelle, my advice is yes, middle school students should follow the same rules. Again, the rules are all about risk, and we want to assess those risks to make sure there's no harm being done. So I know at our fair, we require the middle school students to go through the same procedures that the high school students do. Now they might not be as sophisticated, but it's all the same. We want to make sure that the risk is addressed and how to prevent any kind of unfortunate things that could happen. So my advice is, if you're middle school, yes, follow the same rules that they do for high school, and you'll be safe.
And Henry, we're going to allow that. Oh, go ahead, Joe.
I've actually taught middle school, and as a middle school teacher, I know that risk is a real factor there. So I adamantly adhere to the idea that those forms and those rules should be followed.
Thank you. And we're going to allow that to be the last word because I believe our time is up. Thank you so much for those who have listened, who have posed all of the questions to us. I'm sorry that we weren't able to get to more of them, but we'll definitely be informed by all the questions and try to provide clarity. We will continue to answer your questions via our src@societyforscience.org email account. And so again, thank you to all the panelists who are here with me today. I very much appreciate your input and your sharing our knowledge with the audience.
All right. Thank you.
Thank you.
Thank you.